So thank you for coming to the next in our seminar series of the UK International Coronavirus Network seminar series. We've got two really interesting speakers today. This first speaker is John Kotwa. John's going to be talking about novel beta coronaviruses in white-footed mice in southern Ontario. John's a postdoc working at the Sunnybrook Research Institute in Toronto, Ontario, which, as you know, is in Canada. John is currently researching coronaviruses in key reservoir species, including bats and rodents. His research focuses on integrating approaches from microbiology, epidemiology, genomics, and fieldwork to put together a holistic understanding of infectious disease ecology of emerging viruses at the human-wildlife interface. And John, I think without further ado, people have not come here to listen to me, they've come here to listen to you. So why don't you start? Thanks.
Thank you very much for that introduction. And thank you all for having me here today. I'm really excited to share my work. As mentioned, I look at viral discovery in key reservoir species. And today I'm going to be talking about my work in small mammals, in particular in the Peromyscus species in North America. So just a little bit of a primer before I jump into what I'm doing, because you kind of want to know why I'm just looking in rodents in the forest and in our urban parks. And it's largely because out of the 7 known human coronaviruses, 2 are purported to have originated in a rodent primary reservoir host, meaning they originated in that rodent host, eventually spilling into some intermediate host, and then finally spilling into the human population, where it becomes a predominantly human population or human virus. But it's not just the human coronaviruses that have these origins as well. If we look at other important domestic and livestock species coronaviruses, we can see those relationships as well. So this is a radial diagram expressing the evolutionary trajectories of some of the important human and animal coronaviruses. In particular, at the center of the diagram, you can see that in addition to bats, the rodent host has, relationship to important human and animal coronaviruses. In particular, human coronavirus OC43 and HKU1. I'll talk a little bit more about OC43 because it is included in the beta coronavirus 1 species, which is part of the Embecovirus subgenus. This is a really interesting species of coronavirus because they're all very highly related with high nucleotide identity, but it has a very wide host range, infecting humans, cattle, swine, dogs, and horses as well. So it appears that rodents are a really important reservoir to look at. And because I'm speaking to an international audience, I have to introduce some of the most common rodent species in North America, and that's the Peromyscus species. So they are a member of the Crypsidae family, and they are more distantly related to ones you might be familiar with, like the Norway rat or the house mouse, which is Mus musculus, as shown by that phylogeny on the right-hand side. In North America, the most common Peromyscus species are Peromyscus leucopus and Peromyscus maniculatus, or the white-footed mouse and the deer mouse, respectively. These rodents thrive in a diverse range of environments, from urban exploitation to natural habitats. So they're very good at just existing. In North America, as I mentioned, they have a very wide distribution. This is a map showing the different Peromyscus species. Again, we're focusing on maniculatus and leucopus in this case. In the purple square there, that is where we are located. That's the southern part of the province of Ontario. And you can see within that study area, we have both Peromyscus maniculatus and leucopus, with Peromyscus leucopus occupying the majority of the southwestern portion of the province. So The reports of endemic coronaviruses in their natural hosts in general for wildlife is quite limited. In terms of rodents and small mammals, to our knowledge, no data exists for those species in Canada with coronaviruses in particular. This lack of data on natural virus host assemblages creates quite a blind spot. This also augments our ability to look into relatedness to human and livestock and other animal coronaviruses due to a lack of publicly available whole genome sequences for these coronaviruses. So acknowledging this knowledge gap, we wanted to improve our understanding of small mammal coronavirus diversity in North American rodents and small mammals. I have to say small mammals because we do also include shrews in this, so I can't just say rodents anymore. And so we sought to identify them or detect them, identify them, and characterize, at least genomically, coronaviruses in this host species. So in order to do this, we use the following workflow, starting with sample collection. As mentioned, I engage in fieldwork and collaborate with a lot of researchers who engage in fieldwork as well. So we really wanted to target two different types of interfaces through this study. The first being in Toronto, so the city of Toronto, capital of Ontario. It's a major urban area and the most populated region in all of Canada. This was done in collaboration with researchers at the University of Guelph, my co-lead, Simon Jeeves, and Dr. Claire Jardine. When we focused on urban parks and green spaces in these areas. And then also sampling at Algonquin Provincial Park. This is about 400 kilometers north of Toronto, and it is quite remote. Very little human interface there. And this was done in collaboration with the Ontario Ministry of Natural Resources, specifically Dr. Jeff Bowman, who is currently leading a small mammal project at Algonquin. And this project has been going on for over 50 years now. So it was a great resource to be able to piggyback off of. In addition to Peromyscus species, which were the most abundant species in our sample set, we did also include other small mammals at both of these locations. The extracted RNA from these samples were then tested with a pan-coronavirus PCR assay that targets the RNA-dependent RNA polymerase of the coronavirus genome, the most conserved region of that genome. Because this assay cannot tell us what coronavirus we found in the assay, if we find a positive, we have to follow it up with partial RDRP sequencing. So this takes product from that first round of amplification of the nested PCR and allows us to generate a longer segment that we can then use downstream for identification via blast analysis, as well as enable some preliminary phylogenetic analysis. But the ultimate goal is to generate whole genome sequences for these so we can really dig into these viruses. The challenge with this, though, is that when we're doing viral discovery, there's always a possibility of finding novel, which is great in practice or in theory, but in practice, it's more challenging because a lot of the tools that you can use to generate those whole genomes are not applicable. So we have to use multiple approaches for this workflow. And this includes metagenomic sequencing, which is sequence agnostic, sequence independent single primer amplification, or CISPA, which is also sequence agnostic, amplicon-based sequencing, which requires a reference genome that you then would design primer scheme off of, and then amplify with PCR to then sequence. And then our approach is basically triaging these samples on the quality. In wildlife sampling, sometimes you get lower quality samples than what you might get in a clinical setting. But then also relatedness to other sequences based on that partial RDRP, which is sometimes a gamble. So what did we find? Well, we've actually been able to sample quite a few animals. In the urban parks, we have a sample size of 383. As mentioned, the majority are Peromyscus leucopus, and we found 12 total positives in these animals. 11 were in the white-footed mice, but we also did find a positive house mouse or mus musculus. And all of these positives were from 4 different parks in Toronto. This is a map of the city here, the Greater Toronto Area. And just for some reference, it's about 30 kilometers apart or across right here. So the mus musculus was found at one of the same parks that the other or white-footed mice were positive, and they were found within a day or 2 of each other. From Algonquin, we were able to sample 361 small mammals, the majority being the Peromyscus maniculatus or deer mouse, but we also did have some other species that had quite a few positives, including the southern redback vole and the woodland jumping mouse. So in total, we had 45 positive samples. So based on partial RDRP sequencing, BLAST results, and the phylogenetic analysis, all of the identified coronaviruses here were beta coronaviruses. Now, this is a phylogenetic tree based on those partial RDRP sequences, as mentioned. The highlighted colors, highlighted labels represent the species that sequence was derived from. Based on the clustering of the phylogenetic tree and the BLAST results, which I'll get to in the next slide, it appears that we did find at least 3 potential different coronaviruses. The first I'm denoting purely for the presentation as clade A. This clusters within the beta coronavirus 1 clade, as mentioned, with human coronavirus OC43 and bovine coronavirus, but also they are most similar to Peromyscus coronaviruses from Peromyscus leucopus in the United States. Clade B clusters within the larger Embecco-virus clade, but it is not a beta coronavirus, or they're not beta coronavirus 1s. And these sequences largely came from the redback vole and the jumping mouse. And then lastly, clade C clusters within the beta coronavirus genus, but it is more distantly related to both beta coronavirus or clade A and clade B. Unfortunately, due to the size of this fragment, I really can't make any more inferences, so I'm leaving it at they are beta coronaviruses, and further investigation is required. But I also want to share the BLAST results with you here. This is a heat map generated using the results of BLAST, and the reference sequences at the top are included here because they were one of the top hits for one of the sequences that we included in our analysis. So this includes OC43, the Peromyscus coronavirus from the US, bovine coronavirus, porcine hemagglutinating encephalomyelitis virus. All of these are beta coronavirus 1s. HKU1, and then 2 rodent coronaviruses from Asia. So I'm putting the tree here just to make the comparison a little bit easier visually. When we look at clade A, it's not surprising that clade A clustered so closely with the beta coronavirus 1 species. As you can see, the darker the blue, the higher the percent identity to those reference viruses. So across the board, across beta coronavirus 1s, they're very highly related with some minor exceptions. Moving down to clade B, you can see the percent identity does decrease even to their, what you would say, closest or top hits from those BLAST results. So really for these, there weren't a lot of closely related sequences in the core nucleotide database on NCBI. And then lastly, even more pronounced than in clade B, we see a more distant relationship going down to 77% identity with their top hits. So these are really interesting, and we really need whole genomes for those sequences to really make any further inferences. As mentioned, these clade designations are purely for descriptive purposes and for talking through this presentation. The next I want to talk about, speaking of whole genomes, is our results with whole genome sequencing. We actually had quite a bit of success there. We were able to generate 6 whole genomes via metagenomics from 3 different parks in Toronto, all of which were from Peromyscus leucopus. We were also able to generate a near-complete genome via CISPA from Algonquin. And so this is in Peromyscus maniculatus, which is really exciting because now we have very closely related viruses from 2 different species of Peromyscus. And then also 7 near-complete or partial genomes were generated via amplicon sequencing. So based on the metagenomics, I designed 2 primer schemes from Oak Ridge and Highland Creek and used those to conduct further sequencing on some of the samples, which resulted in those partial or near-complete genomes there. We were only able to get sequences from clade A, so I can't make any other inferences about clade B or C for the remainder of this presentation. Notably, based on open reading frame analysis to look at genome structure, they follow the classic Embecco-virus genome structure, which includes that hemagglutinesterase gene there. This is a pairwise nucleotide identity table, including some important beta coronavirus 1 species, as well as the Peromyscus coronaviruses from the US, just to show how highly similar these are to both human and animal coronaviruses across the board. Obviously, there is some variation throughout the percent identity, as you can see there, but they are all very highly related. Notably, the spike gene is most similar to the porcine hemaggglutinating encephalomyelitis virus for all Peromyscus coronavirus sequences in this study, except those derived from Highland Creek. So Highland Creek was most similar to the bovine coronavirus and was longer than the other Peromyscus coronaviruses that we found and from the US. This is suggestive of 2 divergent coronaviruses, preliminarily denoted as Peromyscus coronavirus A and B. And I'm specifically using the term divergent here just to use as a discussion point, not necessarily insinuating they are actually divergent. More work really needs to be done to look into that. But looking at the whole genome phylogeny, we do see similar clustering as we saw with the partial RDRP tree, which is good and supports previous findings. They all cluster within the beta coronavirus 1 species clade. But as previously mentioned, we have a clade within the Peromyscus coronaviruses that represents Peromyscus coronavirus A, and then also Peromyscus coronavirus B there. But as we discussed on the previous slide with the difference in the spike gene with Peromyscus coronavirus B and Peromyscus coronavirus A, I wanted to dig into that a little bit more. First, descriptively. So I ran a phylogenetic tree of the spike gene and made a tanglegram comparing the topology between the 2 phylogenetic trees. So the whole genome is on the left-hand side and the spike gene is on the right-hand side. And as we can see in the whole genome tree, the Peromyscus coronaviruses all cluster together within 1 single clade. But as we move over to the spike, we have the Peromyscus coronavirus A's still clustering together, but now Peromyscus coronavirus B clusters within a clade that includes bovine coronavirus, human coronavirus OC43. So that identity of these viruses, spike gene to bovine coronavirus, makes more sense, and this topological incongruence between the trees supports that a little bit more as well. Taking that a bit further, we conducted a recombination analysis using RDP4, and we did find a significant recombination event that includes Peromyscus coronavirus B, all 4 of the sequences that we found, to be the recombinant. This recombination event included the spike, but surprisingly also included the HE gene as well. The backbone or major parent of this recombinant was the Peromyscus coronavirus from the United States, specifically isolate 225. And the minor parent was bovine coronavirus. So this event includes 2 genes. It includes most of the spike gene, but mostly the S1 subunit. What was surprising, though, was we also detected in a Peromyscus coronavirus A 1 single sequence, and this was from St. Andrews Park, a significant recombination event that included the HE gene and only a small fragment of the spike gene, less than 1,000 base pairs. Again, similar, the backbone was the paramyscus coronavirus from the USA, and the minor parent was the bovine coronavirus. So I wanted to look at this with the HE phylogeny. This should say HE, not spike. My apologies. But as you can see, very similar to the spike gene, but now when you look at the highlighted sequence here, which was that St. Andrews sequence, it isn't clustering now with the other paramyscus coronavirus Bs. And there's also two of the paramyscus coronaviruses from the United States as well. So really, really seeing a mosaic of these paramyscus coronaviruses that represent almost like a paramyscus coronavirus backbone, and with some changes between them and the other beta coronavirus 1 species, which is really interesting to see. So in conclusion, we provide preliminary insights into coronavirus diversity in small mammals in Ontario. We've identified mosaic paramyscus coronaviruses in both urban and natural settings. The high genetic similarity to known human and livestock coronaviruses warrants further surveillance at this interface, particularly including some more agricultural settings to see if we can pick up on potential cross-species transmission of these highly related viruses as well. And then work is ongoing to generate more whole genomes to further characterize and understand the evolutionary relationships, and dig into some of the other questions that we are asking, like what receptors might these use? What's the host range? Beta coronavirus 1s is a very promiscuous species, as it seems. So it'll be really interesting as this work continues to develop to see what we can conclude from this. And with that, I would just like to say it takes a village to raise a postdoc. Thank you to everybody included in this work, particularly my co-lead, Simon Jeeves. And with that, I just want to say thank you.
John, thank you very much indeed. That's absolutely fascinating. And thank you for your excellent timing as well, which has left a good slot for some questions before Caitlin's talk after you. And I don't know if you want to close the slides so that we can see people more clearly in terms of those that might want to ask any questions. While other people are thinking about it, can I ask you that really interesting to see the role of bovine coronavirus, or so-called bovine coronavirus, in what you've been finding here. And I wonder whether you've got any views or insight into whether this is actually genuinely a bovine coronavirus. It was just first characterized. Maybe it's a spillover into that species rather than from something that's been perhaps maintained in the small mammal population. How much do we really know about the historic roots of that virus? Just too often these viruses get named after the first species they're identified in, rather than more insightful names.
Absolutely. I think some of these findings are really kind of turning our understanding on its head. Particularly, you're right, it seems like maybe they originated in the small mammals and then, like particularly potentially paramyscus species, and then made its way into the bovine population. And then that's potentially how it spread globally, because we have paramyscus coronavirus or paramyscus species in North America, not elsewhere in the world. And it's really interesting to see that the viruses that these paramyscus are harboring aren't closely related to other rodent coronaviruses elsewhere in the world. So we can't really make too many solid conclusions, but I think that this potentially could be the originator of that lineage. And then brings up some very interesting questions about how is this actually happening, right? Like, we were detecting these viruses in Toronto, the city, and we're finding potential recombination with cattle, but there aren't a lot of cattle in downtown Toronto or even in the periphery. So there's questions about how common are these viruses in other bovid species. Like we have white-tailed deer here that are very prominent. They are in the parks that we are sampling from. Could that be kind of an intermediate that hasn't been sampled in depth? And maybe we're just missing that kind of in-between step sequence-wise.
Yeah, thank you. That's really interesting. That's really interesting. And I think so often wildlife hosts have been ignored for these sorts of infections, and just only the species that we can more easily access have been focused on, which thankfully is now changing a little bit, although well, the understanding of the need to change has changed. Funding for it isn't always available.
Yeah, exactly.
That
is a little bit of an uphill battle.
Yeah, yeah. So I wonder if there are any other questions, if people want to put anything in the chat, or if you want to raise just raise your hand, come on screen and ask. I don't see any more questions. There might be time, John, after Caitlin's talk, for people who are who've reflected on things. But thank you very much for a really interesting, excellent seminar. And if everyone could show their appreciation in the standard Zoom manner for a really good talk, that would be brilliant. Thank you very much.
Thank you very much for having me.
That's great.